Yeah, so we're going to get started again for the, so Andy Hoofnagle again is back. And I think now he's going to focus predominantly on plasma proteomics, and I think give you a lot of great applications that they've been working on, the challenges of setting these up and making it work.
So in addition to that, the first part of my talk this afternoon is to sort of put proteomics in perspective in the clinical lab from my perspective, if you will, and then talk about how we've taken that and carried it through some development of assays. But we talked about quality control. We asked a question earlier today. I'm going to talk a lot about validation and quality control from my perspective, what it looks like for the clinical laboratory, and where we'll likely be headed in the future. But first, I wanted to wax philosophical for a little while about what the clinical laboratory is and why we're doing testing using mass spectrometry in the clinical lab in serum and plasma. But so to start, what is the clinical lab? What's it there for? So it's really there to try to help provide a diagnosis, a prognosis, or help in the therapeutic management of disease. And if you think about when you first go into your physician and they're finding out what's wrong, you've got something wrong with you, they first do a history and physical, right? They don't immediately draw blood and send it to the lab. It's an afterthought. So they'll get the chief complaint, the history of what's going on right now, what has happened to you in the past, do you have any risk factors in your family, what medications are you taking? You remember all of this, right? Just going to the doctor, right? Then they do the physical exam. This is what you thought would happen first, but it takes 10 minutes to get there. And then finally, they're like, okay, well maybe I'll send you to the lab to get your blood drawn, or maybe I'll go get an MRI and try to figure out why you have so much back pain. So we're really an afterthought. We're at the tail end of a physician's interaction with the patient. And really, we're only part of a lot of other information that physicians are using to try to make a diagnosis. And when you head into the electronic medical record, and that's been mandated over the past decade, that pretty much if you aren't using an electronic medical record, you're not going to be reimbursed fully by the federal government. So we can actually track the clicks in the electronic medical record, whether you're looking at provider notes, or you're looking at radiology, or search tabs, or maybe even you're clicking on the medications tab to find out what patients are on. Remember, we don't get paid. We're out of business, so we always are looking at billing information to make sure insurance is up to date, etc. And what we've learned is that 60% of the data access, 60% of the clicks that clinicians and care providers click in the medical record are actually lab results, and we only cost somewhere between 3 and 6% of the total healthcare dollars. So in my opinion, we're very, very cost effective. So we in the lab want to provide data that helps distinguish someone who's sick from someone who's healthy. And what that is, the word that we would use to describe that is a biomarker. So that's different than trying to quantify how much drug we've given you to see if you got the right amount of drug. We're going to use these biomarkers to try to pick apart disease from healthy. So here's an example of a biomarker, and you may be able to figure out which one this is when you listen, and I'm completely against using this in a screening situation. But just think about it. The patient comes in for his yearly physical, we draw a tube of blood, the blood test comes back elevated, he comes back for a second draw, it's still high, gets a biopsy, it hurts, he bleeds, he pees blood, he's terrified, he thinks he has cancer. Oh, but the biopsy is perfectly normal. This happens all the time. In fact, 90% of the people who go in for biopsies are completely normal. But that's because we're using PSA as a screening test. And if you think about it, we do this for PSA because urologists have told us it's a good thing, and we could do that for a lot of proteins, if you think about it. We could do a lot of biomarkers if we wanted to. And this is what we're trying to avoid, where the patient says, doctor, I don't feel so good, and the physician says, you look a little green, let's draw some blood. Okay, this is bad, and this is what we want to avoid. And our drive to make awesome biomarkers has to be measured. We have to think about what it really means in the greater scheme of things. So let's think about making a biomarker and how we're going to use it in clinical care. We as the lab directors get to decide what normal is. You may not realize that, but I get to say, I'm going to cut it off here. If you're above that, it's bad, and if you're below that, it's bad. I get the call. And so the way that we generate these cutoffs is doing typically a reference range study where we'll look at the distribution of all the lab results, and we'll say, well, it's abnormal if you're below the 2.5 percentile, it's abnormal if you're above the 97.5 percentile. Now think about that. If I measure 20 things in you, by chance alone, something's going to be wrong with you. Fantastic, right? So this is the definition of abnormal just based on percentiles, which is what we do regularly to define what the biomarker value, what it should be. So there's a lot of science behind biomarkers, and I'm not going to go into great detail, but just to introduce you guys to some terms. When we say the word clinical sensitivity or clinical specificity, that's not analytical sensitivity or analytical specificity. It's literally in a clinical situation. If you have the disease, will it be positive? That's sensitivity. We're shooting for 100%. Clinical specificity is if you're normal, the lab test is negative. Again, we're shooting for 100%. None of the lab tests that we have in the lab is that good. They don't exist. It's impossible. When we want to rule out a disease, we want to have a very high negative predictive value. In other words, when it's negative, it's very, very, very likely that you really don't have the disease. And we want to rule in the disease, we want to have a very high positive predictive. In other words, a positive test means it's very, very likely that you have the disease. And so to put this into perspective, if you want to rule out HIV, we want to be very sensitive. We don't want to let people who have HIV make it through without being told, hey, you have HIV. Now remember, or as I'll show you the next slide, if I might tell a lot of people that they have HIV only to do a confirmatory test and find out that that screening test was wrong. That's the basis of screening. But if I want to diagnose cancer, or if I want to actually rule in HIV, I want to have a very specific test. Any questions about that? So this is what it looks like when you think about chest pain. This is what troponin could look like. So this is a person comes into the emergency room, has chest pain. We run a blood test, and right now we're saying if it's above a cutoff, it's likely that they have had a myocardial infarction, and they should go to the catheterization lab and have their coronary arteries explored. So if I have a lab test that's sensitive, and I set my cutoff right here, I'm going to catch everybody that I want to catch, but I'm also going to catch a lot of people that don't have the myocardial. So it's sensitive, I got everybody, but it's not very specific. Over here, if I put my cutoff here, and this is my distribution now, I'm going to, if it's above this line, it's very likely that you've had a myocardial infarction, but I've missed a lot of people who have one. And this is the trade-off. That's why we don't ever have lab tests that are 100% sensitive or 100% specific. So, the reason that I bring all of this up is the kind of biomarker that you're going to try to discover in your research is going to fundamentally rely on a clinical question that you're trying to answer. If you're trying to identify a patient who has ovarian cancer out of all women who walk into your office, that's a very different question. So we really need to design the best experiment so that we can identify the best biomarker. Because as we've learned over and over again, bad experiments lead to very bad biomarkers. Let me give you an example. So this is the infancy of proteomics. Some of you have heard the story, but not all of you, so I like telling it. This is a paper that came out a long time ago in the Lancet, a very important medical journal. Lance Leodet and Manny Petricoin published this, where they looked at a serum proteomic pattern to identify ovarian cancer. This is supposed to be a screening test. Everyone is supposed to get this test. Draw your blood and see if you have ovarian cancer. And there's different ways that we can try to identify. So we might pick stage 4 cancer, which as you probably know is the most advanced form of cancer versus perfectly normal people. People who have stage 4 cancer are really sick. Really sick. Maybe we'll draw all the cancer cases in 2003 and, we forgot to get normals at the same time, so we'll just draw those a couple of years later. Well, we got all the samples in the lab, but they put them in 5 boxes, the cancer patients, and the normals in the other 5 boxes, but we'll just go through the cancer first and then the second. Okay? All of these are examples of not integrating and randomizing the process. So this red line is from David Ranselhoff. He put the red line in because we can't be responsible for anything left of the red line for a core laboratory. We're just responsible for what's right of the red line. And he basically said, if you don't think about what's left of the red line, you shouldn't be doing the experiment. If you're not aware of everything that they've done before the samples got to our lab, you don't want to be in the business of collaborating with these guys. So make sure you get clarity on that. The way that we should do it is down here. Stage 1 cancer, hard to detect, versus the 9 ovarian masses. This is actually a real live clinical situation. It's hard to distinguish. Where we actually draw up the folks with cancer and normal controls at exactly the same time. And we literally throw the tubes up in the air and let them land on the floor and pick them back up. true randomization, or there's some block randomization processes that Olga could talk about, because she can do it a whole lot better than I can. So this is what the paper looked like. Nobody likes to look at a mass spectrum apparently, so they had to turn it back into a gel. So it's a fake gel. I mean, if you look at it, I hope you guys are projecting well enough. I mean, it's just absurd, right? Look at how awesome that is. So, yeah, so this was really exciting for people, but this was the most amazing table, right? Here, folks with cancer, right? So this is how it's classified by this proteomic pattern. And here are the people with cancer, stage 1, stage 2, stage 3, stage 4. Everybody who had cancer was diagnosed with cancer. Nobody was diagnosed as unaffected. There was nobody who, in these benign conditions, who was diagnosed with cancer with the exception of 3. They discovered this new cluster which was going to change the way we think about ovarian biology. And a lot of people should have asked this question, but they never really did. It was too good to be true. But there were some very talented folks who finally published in the Journal of Bioinformatics. They reanalyzed all the data. And the conclusion is astounding. The ability to discriminate between cancer and control subjects could be based on a mass to charge ratio of 2.8 and 245.5. Uh-oh. Right? There's a problem, right? They had a problem with their data, and they had a problem with how they didn't randomize things. There were big issues showing that there was an experimental bias between the two groups. Could have been anything. Could have been medication, could have been lifestyle. They basically threw up and say, I have no idea what it is. But they recently, or a few years later, came out and explained why they think they got the results that they did. This is what proteomics, this is the black eye that we've had for more than a decade now. Always trying to catch up and trying to beat out what these guys did for us. So, that's history. So what about moving forward? I want to tell you why I want to use mass spectrometry, not necessarily biomarker discovery, which I think is an important process, and I know a lot of people here are doing it, keep doing it because it's important. But we already have a clinical need for mass spec in the lab, and I just wanted to tell you why, because I think it's really important that we're moving in this direction. So this is where I'm intending to scare you a little bit, so that when you go to your clinician and draw your blood, you think about those numbers very carefully, okay? And here's why. So this is the clinical immunoassay where we're looking for TG. I showed you this before, where we're making the sandwiches and we're looking for the reporter antibody. But the big problems with this kind of assay are things like specificity. These antibodies might actually recognize some other protein, right? It's possible. There's related proteins in plasma, and these antibodies aren't always perfect. The standardization, I showed you all those calibration. I showed the example of IGF-1. I have some more examples. There's something called the hook effect. If you put in so much thyroglobulin into this assay, you'll saturate all the binding sites of the captor antibody and all the binding sites of the reporter antibody, and you'll get no signal because you can't make a sandwich anymore. So you get a false zero. That happens. There's a bunch of different clinical situations where that happens. Anti-reagent antibodies can actually bridge the gap between the reagent antibodies and get a false positive. I'll show you an example of microplots. And if you think about it, immunoassays are inherently single-plex. There are some examples of assays that say that they can measure multiple analytes at the same time. I'm not going to show you data today, but there's really big problems with multiplex immunoassays. So all of these, I think we can fix with mass spec. I propose to. So let me just give you a couple more examples. Again, this looks a little bit faded, I'm sorry, but hypothyroidism, we diagnosed with thyroid stimulating hormone and TSH. We usually follow it up with some free T4 measurements, but there are a lot of primary care physicians who would treat if you have an elevated TSH. That's just what they learned in medical school, and that's what they do. So these are actual data from Bill Roberts, who recently passed away, a little bit too young, showing that different manufacturers' results, if you send samples to laboratories who are running these instruments, will get different results. And one of the cutoffs that's been proposed by endocrinologists is 5. So if you're above a 5, some primary care physicians will give you drug. If you're below 5, you won't. So if you look, these are 3 hospitals that are half a mile apart in Seattle. So you can conceivably go and get therapy at one place, and at the other 2 hospitals walk away with nothing, all based on a lab test. all based on FDA-approved lab tests. I talked about the autoantibodies causing false negatives this morning, so I don't need to dwell. I do want you to realize that we know about this problem for thyroid globulin because people have been studying it, but lots of people with cancer, especially breast cancer as an example, make autoantibodies to proteins. And we haven't even explored how these autoantibodies interfere with those immunoassays because it's just hard to do. So we've never done it. So I give the example of thyroid globulin, but lots of assays used in cancer patients likely suffer from this problem. This is the example of microclots. Microclots are these weird fibrant strands that are able to grab onto the capture antibody and the reporter antibody and give you a false positive. So in plasma, we don't actually this is true. It really was. In plasma, we actually detect beta-HCG, which is the pregnancy hormone, one of the pregnancy hormones, whereas in serum it doesn't exist because serum is completely clotted. We get rid of all those microclots. This is not microclots, I want to go. but it is somebody from our laboratory where it was a false positive HCG. It can't be real, but here's what happens with immunoassays. So, it doesn't happen in serum. So, just to summarize these kinds of things, and summarize the impact of these, if we don't have good agreement between instruments or different manufacturers or different assays, when we write guidelines and we say that there's a cutoff, that number is really only valid for a single platform, whatever platform we use for the study. This also means that folks with cancer have to be followed by the same assay over time. Because if I switch, all of a sudden I might have twice as much biomarker as I thought I should, and everybody goes crazy. We can go from one place to another and have results of so for 30% of patients who've looked, in some cases, many patients actually correlate well between two platforms. But then there's like a subset that makes this weird cloud. They just don't correlate. No one knows why. Different epitopes, different interferences, no one's really sure. The autoantibodies, so I mentioned, again, lots of patients with cancer have autoantibodies. It's actually a good thing to have autoantibodies to breast cancer antigen. It's actually a good prognostic indicator. We have no idea how it interferes with our assays. Unfortunately, patients with autoantibodies get more diagnostic studies because the physicians know that they can't trust our assay. And unfortunately, we have a delay in diagnosis. We can't detect that thyroid globulin or some of the other things as early as we'd like to. These are the worst anti-reagent antibodies. We get non-specific false positive results that end in more radiation therapy, more chemotherapy. Women have had their uterus removed because they thought that they had uterine cancer. That's actually happened here at UW many years ago. Bad things happen because of anti-reagent antibodies and proteins. Okay, any questions about that? I wanted to scare you just enough so that you believe me that we should really be doing mass spectrometry to measure the proteins in the clinical lab. It's a great conversation to have with your physician, I thought. Okay, so that's all about the clinic, but what about in just everyday life? Why would we want to quantify protein and complex mixtures? There's lots of basic science. We know that we have a protein that we're interested in quantifying. We can't find a good antibody or a good Western blot. Can we use mass spec to quantify the proteins just in our simple basic science experiment? Discovery, we want to catalog the proteins and maybe discover some biomarkers that are different. Maybe we can use it to select patients for clinical trials. This is something that the pharmaceutical companies are very interested in. Can we use biomarkers to establish toxicity of novel drugs? What about efficacy? Can we use proteomics for diagnosis, prognosis, or therapeutic management in the clinical setting? So these are all possible reasons. I will say I like to measure proteins by mass spec because it's fun. So let's think about the experiments. In basic science, we may have cells, we may have animals, we might have a few human samples, not thousands, or we might be doing discovery. What we expect in these kinds of experiments is for trends to repeat. We might expect to see high peroxidase 3 in one set of human samples versus another. But if I did this experiment the next time, maybe these lines would be a little higher or a little lower, but we expect to see decrease. That's what basic science is all about. Expect the trends to repeat, but not necessarily the numbers. Now, some people will take issue with that, and I don't mean to offend anyone, but that's, in theory, what basic science is all about. When we start to do retrospective human subject studies, again, we might be doing some discovery experiments where we're taking one rack of human samples that have been frozen for a year that are normal and another rack that have cancer to try to find new biomarkers. We might be trying to verify biomarkers that we've seen from our cell-based discovery. There's this term called qualification, meaning you want to qualify the biomarkers in some way to help somebody believe. This kind of experimentation may not need IRB approval. It might, but be careful. But most likely it doesn't. And I call this freezer diving, where you just go in and you're getting samples that somebody else collected. You had no responsibility for collecting them. And then there's this idea of a prospective clinical trial, where we're involving patients and we're trying to demonstrate that our assay is useful in the diagnosis, prognosis, or management, or we're using these kinds of assays to dictate what kind of, which side are you on, to randomize patients, etc. And I just want to stress that we need IRB approval for these kinds of studies. And if you need IRB approval, you should be fully validating your assays. And really, this is now using our novel biomarkers in patient care. I just want to stress again, please fully validate an assay like this. The FDA gets word they haven't fully validated, they will shut you down and cause problems for the rest of the field. So, when I say validate, what do I mean? There's a lot of definitions of what the word validate might mean. I have a very simple definition, which is a sincere attempt to demonstrate the robustness of your system. You're going to stress the system. You're going to try to break it and prove that it's not breakable. Okay, that's what I'm defining as validation. So, if you think about the parts of the immuno-MRM assay, that's the immunoaffinity enrichment of peptides with antibodies, just as an example. It starts all the way back when you draw the blood. Everybody have their blood drawn and someone's like, okay, now pump your fist. Bad. They leave the tourniquet on for like 4 minutes. Oh, I'll be right back. I have to go get us something. I'll be right back. And then they're gone. They come back 4 minutes later. You've had the tourniquet on for 4 minutes. Bad? Does it happen? Maybe it hasn't happened to you. It happens all the time. Whether you're laying down, sitting up, can change lots of small molecules and proteins and what we draw from the tube. Which tube did you pick? So there's a purple top tube, which has EDTA. There's a blue top, which has citrate. There's an orange top, which has an activator to actually cause clotting to go faster. There's a gray top. There's a tiger top. There's so many different kinds of tubes. It's unbelievable. And I have to be honest with you. EDTA and red top, they always make the assumption, hey, I've got a lot of plasma, EDTA anticoagulant plasma, and you do a bunch of proteomics studies in it, and then you switch to serum, and you get a totally different profile of what's in the tube. That's true for both small molecules and proteins. So just picking the blood draw tube is important. How to store it? Was it sitting on ice for an hour before I got into the -70, or was it on ice for an hour and then in the 4-degree refrigerator for 5 days and then into the freezer? What happened to that sample before? And is it important when I take care of patients? We talked about pipetting. What tips did you use? Were they low bind or not? What kind of tube did you use? Is it a half mil or a full mil? Because the transfer of heat is very different. Internal standards. Is it the batch that I got from the place in China, or is it the one that I got from New England Peptide? Are they the same? I don't know. Talk about denaturation, reduction, alkylation, digestion. The magnetic beads, different batches of magnetic beads from Dynow are actually quite consistent. There are other manufacturers of magnetic beads that are coming onto the market that I think we need to be more careful of. Different antibodies. So you guys have heard the story of the golden rabbit, where the rabbit dies and a whole assay disappears. That happened to Siemens about 5 years ago. Literally, they relied on the rabbit's dead. No more assay, and they couldn't make it again. So washing and elution. How am I including some detergents in my washing step or not? Am I including an acetonitrile and acid, or just acid? And then with auto sampler, HPLC pumps. We're going to go into a little bit more detail about those steps as well. So unfortunately, all of this can make your assay go bad. Any one of these steps can make it just crap out. And that's what we're going to try to validate, is to try to figure out which of these steps actually can cause problems, to make sure that we try not to. So I've defined it as a sincere attempt to define the robustness of the system, but places like FDA and CLSI and the Centers for Medicare and Medicare Services, the College of American Pathologists, Washington State Department of Health, New York State Department of Health, they actually have guidelines that they want you to follow when you're validating an assay. So in a regulated environment, we have to rigorously define the performance characteristics. We ourselves get to say how good we think it should be. That's what's amazing about the FDA. When you walk in with an application to the FDA, you basically say, I think my assay should have a CV of 15%. They're like, okay, cool. Show us that the CV is 15%. That's it. Oh, I think it should be 23%. Cool. Show me this 23%. As long as you can, your assay gets validated, which is amazing. But the documentation, the stacks and stacks of paper, you sit down and you say, I'm going to make an assay that can do this. So you write it all down and you sign it in the data and you put it aside. Like, well, I've thought about it and I really think that I should do this little tiny thing and you sign it in data. And then the next time you do the assay, you sign in data that actually you did that change. So every time you want to make a change, even in your development process, you have to write it down. So the paperwork gets really, really, really long. Transparency, in other words, you, the end user, you never get to see that. Because transparency doesn't matter. FDA, they're the ones, they get to decide. And if they're happy, that's all that matters. You will never see the raw data that led to an FDA approval or clearance, as they like to say. Unregulated, more like publishing guidelines, right? We want you to include the following things. Funding agencies, I think NIH is a bunch of wimps when it comes to this. They never actually say, you should do this because it's best practice. Instead, they rely on their peer reviewers, both in journals as well as looking at grants, to decide who's actually doing a good job and who's not. Most importantly, when we're training our graduate students and our postdoctoral fellows, we can tell them what good practice is. This is what you should do when you run an assay, so that when you get the results, you're confident that you've done a good job. So I think we are probably the most important people to help ensure good validation. And in the unregulated environment, transparency has to be a fundamental thing. If you're not telling people truthfully and clearly what you've done, we'll never know if you did a good job or not. So a couple of publications came out a couple of years ago. The first one, Henry Rodriguez from NIH, part of this, he's basically the program director for the CBTAC program, and Steve Clark got together to have a basically a workshop to think about fit-for-purpose assays. And we ended up breaking things down into 3 different tiers of assays. I know it's small, I have it blown up in the next, but Tier 1 being clinical assays, Tier 2 being clinical research assays, and Tier 3 being discovery. So if you break it out and you think about those experiments that we were doing before, and you start with your discovery, doesn't have to be a super precise assay. Again, we're looking for the big magnitude changes because we want these biomarkers to be, they're so, night and day. We may have gazillions of amyloids in this kind of discovery process that we're doing. Tier 2, we're still doing research, but now we're going to start to include things like internal standards because we're really looking for some precision. We want the trends to be repeatable, but we may or may not care about what the concentration is. And we don't care if we actually get the same value a year from now. We just want to repeat the trend. And then in Tier 1, we're making clinical diagnoses here. We want a highly precise assay with QC. We want the values to be the same over time. And generally speaking, as you go from Tier 3 to Tier 1, the number of amyloids that you have in your assays is decreasing. So this is a general framework within which to think about validation. And so building upon that, we in the CBTAC program tried to come up with some concepts around how would you want to prove that your Tier 2 assay is really worth anything. And so we put in 5 different experiments that went from defining the lower limit of detection and showing some initial assessment of precision. So again, linearity and precision. And then we had some other experiments which I think people felt were a little bit over the bar. But at least the first 2 experiments have been included in the CBTAC it's a web-based portal for assays. And I'm going to talk about it more at the end, so I won't dwell too much. But there's actually a bunch of assays that have had those experiments performed on them available online. And again, it's searchable, and I'll talk about it in a little bit more detail later. So Russ Grant is at LabCorp, a very talented analytical chemist who's done an amazing job increasing the throughput. He can do 2,000 samples in a day. Hold on, think about that for a second. On a mass spectrometer, you can quantify proteins in 2,000 samples in a day. That's amazing.
One mass spectrometer.
On one mass spectrometer. One mass spectrometer. Granted, there's 4 HPLCs, but still. Amazing stuff that he has been able to do. So he and I spent a lot of time, years, and discussion coming up with what we thought would be reasonable experiments for people who wanted to publish the assay that they're going to use or have used in very large clinical research studies. So again, the linearity and reproducibility being the hallmarks, the lower limit of quantification, so just doing some dilution experiments to show how low you can go, adding potential interferences. So for us, in the clinical lab, that's things like patients with liver failure, patients with kidney failure, patients with very high cholesterol, triglycerides, those kinds of things to make sure that they don't interfere with any of our digestion steps, etc. And then the stability, stressing the digest and showing that the peptide is stable, but also stressing samples before we do any digestion to show that the proteins are stable. So these kinds of things, I'm going to show you what I mean by this in some of the other a little bit later. But the thing that we found super interesting is that the peptides do degrade. And as you increase the amount of trypsin that you add, the degradation happens faster and faster. So I have some pictures of that I wanted to show you too. So again, this is what we think is necessary to validate protein quantification for publication. So if you submit something to clinical chemistry, which is I'm one of the editors there, then I require that all of these experiments be performed. You can actually do this in 134 injections on your mass spec, just so you know. This isn't extraordinary, but if you're going to use it in thousands of samples, 130 isn't enough. Taking it one step further, if you really want to develop something for the clinical lab, I encourage you to read these documents from the Clinical Laboratory Standard Institute, or CLSI. They do cost money, but it's possible that your libraries have them already. These guys spell out very interesting experiments, best practice, next to best practice, all for the purpose of either submitting to the FDA or for making an assay that's good enough for the care of patients. And most recently, we put out the CLSI, and I was on the rating group, published something called C62A, which is a guideline for the development of an LCMS method and deployment. There's no talk of proteins in this yet. We're getting ready to write that document now. But this actually outlines all the experiments that takes all of these and puts a little LCMS twist on them. So it's a great read. It's thick. But it gives a lot of good guidance in trying to make the best kinds of assays. So I wanted to talk about QC in a little bit more detail about some of the validation experiments that we have done. First, let me talk about quality control. There's different levels that you can think about quality control. There's the instrument. There's QC that we can do with each of our samples. We can do QC around the digestion, and we finally have batch QC. So we talked about how something can go wrong with our assay. Just the mass spectrometer, right? Our mass calibration has been good. We can see a drop in sensitivity. And then if you stop and put your thinking cap on and think about, well, what can go wrong with my mass spectrometer that affects mass calibration and sensitivity? There's lots of places. There's the source, there's all the lenses, each of the quadruples, the collision cell. The vacuums can go bad or not be performing as well as they should and letting in a certain amount of gas. Detector, that can go bad or behave very funky. Circuit, I mean, all of these things can go bad, and unfortunately you don't have any idea which one's which. We really have to look at the larger picture. When we think about chromatography, we can watch retention time shifting or peaks get broader, we can start to lose resolution. And what is it? The first thing we often do is change the column, because it's the easiest thing. Well, that didn't work, and now we have to do our pressure test on our pumps. So the column oven says 50 degrees, but it's not acting like it's in 50 degrees. Things get clogged, things get dirty. Lots of things can go wrong on our mass spectrometer that can affect the results that we have. Now, if you have, I'm just going to stop and think about this for a second. If your retention time shifts by a minute, is that the end of the world? Who knows? unless you validated putting on a slightly longer column, a slightly shorter column, or mess with things to see how your retention time shifting affects the interferences or your ability to quantify things precisely, you don't really know. So one of the things that we do is we have a window within which the retention time must fall. If it goes outside of that, it flags it. And depending on the amyloid or depending on the assay, we may have, either you have to repeat this whole batch, you may have to repeat that one sample. We've tried to figure out how these things can actually affect the quantification that we're trying to do. So to keep going, what about an auto sampler? I don't know if we pays attention to carryover, but in the clinical lab that's a big problem because there are patients who have hundreds of times more than they should. And then when the next person is supposed to be normal, well, oopsies, we actually have a little bit of carryover. Now they're positive, and they never should have been. And we have to make sure that carryover is a problem. Air bubbles, so we don't actually pick anything up in our auto sampler. Sometimes we don't make a complete injection, we just a leak somewhere, things can get contaminated. And again, we have hardware that we can blame, and unfortunately it gets into swapping things out. But we want to try to figure out where our assay can break. I mentioned some of the interferences that we do in the clinical lab. There's also a lot of exogenous substances that might interfere. We have to think about matrix in general, and then of course analytical. Are you getting polymers that are interfering with things or not? And then of course these kinds of things will lead to errors in our quantification due to ion suppression that we're not ready for, or inappropriate signal either on our internal standard or in our endogenous channel. So interferences can be a problem. So this is where, so this part, right, when I have to make my calibrators and my calibrators go bad, either I've made them wrong or something's unstable. Data integration, that's huge. If you go into our clinical lab and you walk over to somebody who's actually running the mass spec, the first thing they'll do is grab the mouse and change the integration on the key. They're just not happy. They're just not happy with what Skyline did or with what Waters did. And they'll immediately grab onto that little tiny box in the bottom right-hand corner and fix both sides so the key looks beautiful, at least in their eyes, right? You're not supposed to do that. The FDA doesn't like that at all. We do it and everybody does it in our lab, so there's nothing I can do about it. But this is all about software or people making errors. And so we've actually went through, Skyline is pretty interesting. Skyline, you can look at smooth data, not smooth data, or data smooth using a different algorithm. So there's two different algorithms. And unfortunately, when you log onto your computer and look at it using one smoothing algorithm, but your friend on the other side of the lab uses the other, and you take your data and you walk over and you plug it into his computer, you'll actually be looking at it with his smoothing algorithm. And you'll look at the integration and be like, what the hell? Who did this? Like, what did you guys do? But really, the data, the actual integration is being done on the raw data. And the integration is actually, the edges of the integration are being defined during those smoothing steps. So the raw data is really what you're integrating, and that's probably the best place to look if you want to review the integration. But we actually compared a person who was sitting in front of this Fizy-Golay and somebody who was sitting in front of the second derivative, and when they started to play with the integration, it was wildly different. And the precision of one was horrible, and the precision of the other was fantastic. So just keep that in mind. Don't change the integration in Skyline just because it doesn't look right. Make sure you understand and be consistent from day to day. Okay, so to sort of summarize what we're trying to go for quality control, I will reiterate that assay validation should be a sincere effort. When we want to test our system suitability, we want to figure out if the instrument is working before we ever touch a patient sample. Before the mass spec ever touches a patient sample. We can do a couple of different things, like looking at unextracted materials, to try to make sure that the instrument is as sensitive as it should be. We might digest blanks, in other words, a sample that we know doesn't have the protein that we're interested in, to make sure that we don't actually have contamination from carryover somewhere else in the system. For the batch process quality control, we're going to make sure that something goes all the way through the system. In other words, it's going to be a sample that's just like my regular samples, and I'm going to carry it all the way through to make sure that the numbers at the end of the day fit into the bracket that I've defined for that. And ideally, we're going to do this more than once throughout the batch. Not just once at the beginning, but once in the beginning, once in the middle, and once at the end. To make sure that all the way through the 96-12 plate, everything went okay. So these could be, I could put in matrix match samples. I could include quality controls that are near the medical decision points, which is mandated by CLIA. So there's different things that I can do for quality control. But the thing that sets us mass spectrometrists apart from those people who use immunoassays, I could do that too, but anyway, so the things that set us apart are our ability to look at the internal standard peak area to make sure that ion suppression isn't too severe. Retention time, we now can be confident that this is the molecule that we're interested in, especially because we can look at different transition ratios. We can be confident that the fragment ratio is meeting what we think it should. This we can do in every single sample, and we should do in every single sample. Something that you just can't do in an immunoassay. So I just wanted to give you some examples of some of the QC that we use. This is awesome. This is a quality control sample that's taken all the way through. So we're adding internal standard peptide, we're digesting, and then we're injecting it onto the mass spectrometer. Actually, sorry, the internal standard peptide was expressed inside it after digestion. And so we start back here, and after a few weeks, the signal decays until less than 10% of what we started with. Holy cow. This is happening in your mass spectrometer at the top. Especially if you're doing nanoform. Your instrument gets dirty very quickly. So then the question becomes, well, how bad is this? Is this really bad, or is it just a little bit bad? Is this a useful parameter for us to use for quality control? Should this be staying way up here within some 20% band, or is it okay that 90% of your signal's lost? We can, this is another way to look at your instrument, and I'll answer that question later. So this is a system suitability that SciEx wants you to use on your, this was the Q-trap back in the day, and this is the peptide that they wanted you to monitor. So it's this nice big blue peptide in the middle of the screen. It's always so big and delicious and juicy. But there's another one just to the left of it. But if you look at that, you'll actually see that reduction. The same thing that we saw in our QC sample. This is lovely. This, not so good. So what does it mean? So this is in the control sample. This is the peak area response of one of our analytes. And you can see that as we start to lose signal, we actually started to get the wrong answer. The response ratio at least was incorrect. When we use the calibration, so we're going from the response ratio to the protein concentration, we have the calibration, we actually see that it's not exactly right either. It's within 20%, which is nice, but we're starting to get some changes. So going back, there's this huge decay, but there's no obvious break here. We have something sloping up, but here we have this disconnect. So, yes, we did something that seems to fix the assay. In fact, it was just changing the capping column. It didn't change the total amount of signal that was coming into the system. And you can see that we started to have the trend again right after we changed the capping column. So, how much did these kinds of things affect? How much did losing 90% of the signal affect this assay? We were still within about 20% of the correct value, which may or may not be good enough for the application that you're trying to do. Something else that we were looking at, so there's 3 different transitions, and so we can calculate 2 different ion ratios. We monitor this over time. You can see that this ratio is very stable. This one is more scattered, but you can also see a little bit of a trend there. You can, when you actually, so this is actually in the QC material. You'll see that as the instrument started to lose more and more signal, one of those ratios actually started to get much more variable than before. So we can program into our QC we can put into our QC program, looking at the precision of the transition ratio across the batch to make sure it is as good as it should be. So another type of QC that you can include in your assay. And then lastly, the thing that I would leave you with is the benefit of having that internal standard protein of ApoA1 is we can actually now spike that into N15ApoA1, we can spike that into every single sample. And the ratio of one peptide within that N15ApoA1 to another should actually be constant. And we can make, again, we can make a QC parameter out of that. If it falls outside of that, something went wrong with the digestion, we need to repeat that sample. And so you can look at this peptide ratio over time, looking at each of the samples. So if there were something down here or something up there or whatever outside of the band that you think is okay, you can repeat that sample. The kind of quality control that you can't do in an immunoassay. So, thinking about doing the batch quality control, it's not the same thing as the process quality control that you might read about in some textbooks. Normal process quality control is over time. I run a QC material at 8 a.m. and 8 p.m., 8 a.m. and 8 p.m. to make sure that everything's going okay. If you try to do multiplex QC, so remember that when I told you that you could be abnormal when I measure 20 things in you, we have to set some standard deviation around which we're allowing our QC parameters to flex. And the more quality control parameters we have for more and more analytes, more and more chance that you're actually going to be out of control in your assay. And you have to decide what you're going to do with each one of those. And it's an interesting thought experiment, and practically speaking, it can really change how many repeats you do. Whether or not you have native calibrator and QC materials, it's always best to have your quality control materials be the same as the samples that you're measuring. And I will tell you, again, without good calibration, long-term quality control is simply not possible. Okay, so I'm going to switch to an example, unless you guys have questions. So the one that I'm going to talk about is an assay that we developed and validated for vitamin D binding globulin. This was our the first assay that we developed to actually try to meet those criteria that Russ Grant and I wrote in that paper in clinical chemistry. So why vitamin D binding globulin? So vitamin D does a lot of things in the cell. This is 125-dihydroxy vitamin D. Makes its way in to bind to the vitamin D receptor, which has a heterodimer with the RXR receptor and leads to the transcription of lots of different proteins. A lot of them are related to regulation of calcium and phosphate in the body, regulation of bone development and maturation, but also turns on its own destruction, which is the 24-hydroxylase enzyme, and even controls how much renin is being expressed in the kidney, which is directly related to your blood pressure. So vitamin D could potentially have lots of effects outside of the blood, out of the bone health. And so one of the things that we became interested in is does vitamin D actually have a role in cardiovascular disease? So as an example, we know that vitamin D regulates immune cell function. It regulates the activity of beta cells in the pancreas. It regulates vascular smooth muscle cells in the renin, aldosterone, and angiotensin system. And it even regulates the differentiation of cardiac myocytes. So if you think about vitamin D having all of these different actions, if you ask the question, could vitamin D be related to cardiovascular disease? The answer that we came up with was yes. And so we were able to get funding to take a look at vitamin D in the context of cardiovascular disease. And just to put this in, all of this into perspective, this is the National Health and Nutrition Examination Survey. This is a little RV that drives around the United States drawing people's blood. It's funded by the CDC. So they draw blood in the south during the wintertime and in the north during the summertime. So they're always warm and cozy. And when they drive around drawing people's blood, sometimes they stop and they basically look at 4-year blocks. They try to figure out how we are doing from a health perspective. And what these data showed was that we have a huge problem with vitamin D deficiency. So the cutoff for being vitamin D sufficient is around 20 nanograms per mil in most laboratories. So if you look, black males less than 30 years of age, half of them are vitamin D deficient. This is black middle-aged females, 75% of them turn out to be deficient, depending on how you calculate it. What you, the population that you're looking at, and this one's about 75%. So there's this huge problem with vitamin D deficiency, but you'll notice that actually whites have a lot more 25-hydroxy vitamin D in the plasma. And so that was a bit of a paradox, because even though blacks have less vitamin D, they have stronger bones. They don't break their bones like Caucasians do. So there's this big discrepancy, big debate about why do blacks have less vitamin D than whites, yet they have stronger bones. So one of the hypotheses that was developed in this New England Journal of Medicine article has to do with bioavailable vitamin D. So let me tell you a little bit about that. This is data from their original population, and what you can see is that the distribution of the blacks shows versus the whites is that there is a lower concentration of 25-hydroxy vitamin D on average. So vitamin D binding globulin is a protein that binds vitamin D and transits it throughout the blood, and vitamin D binding globulin binds onto vitamin D so tightly it's thought that it never gets off to actually do its business. So there was a hypothesis that even though blacks have lower concentrations of 25-hydroxy vitamin D, the hypothesis was that they have less vitamin D binding globulin, which would actually allow them to have the same amount of vitamin D that was available to actually do the business. And so the folks from Ravi's lab at Harvard used this R&D Systems monoclonal immunoassay to quantify the amount of vitamin D binding protein, or vitamin D binding globulin, in the same population. And what they found was that whites had this nice distribution of vitamin D binding globulin, but the blacks actually had this bimodal distribution. And as you can see, on average, blacks had lower concentrations of vitamin D binding protein. And when you then did the calculation, then the calculation uses 25-hydroxy vitamin D, albumin, vitamin D binding globulin, and some equations that were determined back in the 60s and 70s. You can calculate bioavailable vitamin D, and voila! Your bioavailable vitamin D is exactly the same in blacks and whites. Problem solved. This was huge. This was a great paper. People are so excited about this. Don't even have to measure vitamin D ever again. We know why it is that. But if you look in the supplemental materials, there's some interesting data. So here's a histogram of the vitamin D binding protein concentration that they observed, broken down by these isoforms, which are genotypes. So there's three haplotypes, GC1S, GC1F, and GC2. These are named for how fast they migrate on gel electrophoresis. It's actually agarose gel electrophoresis. This one's fast, and this one's slow, and that one comes second. So, and these polymorphisms are very different between Caucasians and blacks. Blacks have a lot more GC1F and GC2 than whites, who are predominantly GC1S. Wow, that's kind of frightening, isn't it? And so Dan Bickley and Bruce Hollis published this, which was a letter to the editor about this. The monoclonal antibody discriminates between GC1F and GC1S variants to provide artificially low values for vitamin D binding protein. This is a hypothesis. But they got to say this, and thus bringing the conclusion of this study into question. That's the kind of paper that we want to be able to write. Like, that's possible. So this was their hypothesis. We had the same one. So the question was, if we developed an LCMS assay, could we show that in fact the monoclonal antibody was discriminating against them, and that in fact blacks and whites have the same concentration of vitamin D binding protein? So to do that, we had to select peptides, and we used a full skyline process to do this, where we identified how many peptides had more than two amino acids, tried to think about what the sweet spot was in terms of mass, so we selected peptides that had between 6 and 25 amino acids. We started to exclude peptides that had non-ideal amino acids. You have to decide if that's really appropriate for you. We decided to leave out methionine and cysteine and tryptophan, just as an example. We then looked at the peptides that had good precision by injecting the digest with the purified VDBG over and over again. Which peptides were stable to freezing and thawing of the digest, and showing that they didn't lose signal. Again, beating it up and make sure that it's going to exist. And then peptides that have good signal intensity in plasma. Finally, we did find some chromatographic issues as we started to look in actual patient samples, so we narrowed it down to 7 peptides that we carried through the rest of the experiment. We ended up throwing one of those peptides out for other reasons, and made 6 internal standards, 3 to quantifying peptides. This peptide turned out to not be as stable as we thought it was, even after doing all that beating up. It loses intensity over time, unlike the other 2. So we have 2 quantifying peptides that we're using to calculate the concentration, one that we're using as quality assurance. And then we have peptides that actually can tell us what the genotype is of each of the samples, which I'll talk about more at the end. So the chromatography, this is a pooled sample. It's a bunch of different samples, a bunch of different human samples pooled together. And so you can actually see the peptides that correspond to the polymorphisms, as well as the three quantifying peptides. Very good chromatography with very little interference. And again, I'll remind you of these kinds of experiments. We wanted to show that this assay was robust by using the experiments that Les and I had described. So the last thing that I didn't really talk about before is this transparency thing. So providing the raw data, not an Excel spreadsheet that shows peak areas, but the actual raw data. And how can you show raw data? You can upload a Skyline document. So we actually have a Skyline document from the whole validation experiment sitting on clinical chemistry that anybody can download and look at, but in the comfort of their own home. And it doesn't, you don't have to upload, Excalibur, you don't have to upload, you can put it all into this vendor-neutral software, which I think is a really great way to let people know that they can look at your data and decide if they agree with what you said or not. So let's first talk about this reproducibility experiment that's 5 by 5. So in this experiment, we have 5 different days, and we're running the same sample in 5 replicates on each of those 5 days. Okay, so it's 25 digests spread over 5 days. And the way that we do the math is we look at all of the replicates in one day and calculate the variability for that one day. And then we pretend that all of the replicates here were run as the same sample. This is the same sample. We look at the inter-day variability. And using the sum of squares, or you can do this using ANOVA as well, you can actually get what the total error is, which is now an estimate of, if I ran the sample today, how different do I expect it to be if I ran it a couple of months from now? The math is here, so hopefully you guys can see it on the PowerPoint. And we want to do that at a couple of different levels and compare the total error. And this gives us an idea of how robust our experiment really is. So I mentioned Chris Schubert, he was at University of North Carolina, who's now at LabCorp. He published in MCP this concept that you could actually model the rate of formation of endogenous peptides and the rate of decay and come up with the actual concentration of the peptide. I'm glossing over a lot of what he did. But what we learned from that is if you add a lot of trypsin, you generate your peptide and it slowly disappears until it reaches steady state. Probably when the trypsin is dead, which in this experiment shortly after 2 hours. At the same time, the internal standard that you spiked in before the digestion disappears very quickly as well. So when we look at the peak area ratio, or the average of the 2 peptides in terms of the peak area ratio, things actually look very stable after about 15 minutes. Which is pretty cool. So you can digest things very quickly, provided you add a lot of trypsin, and provided you include your internal standards beforehand. And from this data, so again, we're adding the internal standard before digestion rather than after. When we do this experiment and look at the peptide peak area divided by the IS compared with the calibration curve, you lose 37% of your peptide even in a 2-hour digestion. Pretty amazing. So think about that when you're designing, and we're building this off of the Schufer paper from MCP. So another thing that we are interested in is this concept of linearity. Again, linear and precise makes a good assay. The concept of parallelism is asking, do I have any matrix effects? In other words, if I have a protein sitting in fish serum, does that protein behave in fish serum the same way that the protein behaves in my patient sample? And so what we do for this kind of study is we start to mix things together. You'll see the black dots are pairs of patients that we've mixed together in various concentrations. If we take a patient sample and mix it with chicken serum, we want to make sure that we're getting the same response, the expected concentration that we would. And then finally, if we spike recombinant protein into our sample and then dilute it out with chicken serum, all of these mixes should lie on the same line. That's the concept of parallelism. What this says is my recombinant protein is behaving like my patient protein, my chicken serum is behaving like my patient serum. Things look pretty good in terms of matrix effects. So this is my favorite figure that I've ever published. I got to put it in color. what this clearly demonstrates, here's the immunoassay. This is that that R&D immunoassay that was used in that paper. Here's LCMS, and you can see the very clear genotypic effect of the relationship between the immunoassay and the LCMS. The manufacturers of the immunoassay actually donated the ELISA plates to this study, and they have since taken it off the market because it's a bad idea. So, this is fun, and this is how LCMS can show that immunoassays are having big problems. If we actually plot now a distribution using the R&D immunoassay in blacks, it looks just like that paper that was in the New England Journal of Medicine, this side. But if you actually look at LCMS, there is no different distribution of vitamin D binding globulin in plasma. It's just that simple. So, the other question that we had was, could we determine the phenotype and how accurate would we be? So we can get the genotype of all these folks. But what if we digested, took the peak area of these polymorphic peptides, divide by the internal standard peak area, because we had internal standards we were adding before digestion. Take the polymorphism peak area ratio and divide it by the quantifying, and come up with some empirical cutoff to define whether or not the polymorphism was there or not. So empirically, we used a lot of data to establish this is where it doesn't agree and this is where it agrees. So it was easier for these two genotypes than for this one. We've since gone to a more sensitive mass spectrometer, and the ability to distinguish zero from one presence of an allele is much easier for this now. But this is what we were able to publish a couple of years ago. 2 alleles, 1 allele, 0 alleles, 0, 1, and 2. This turns out to be 1, and the rest of it is all 0. So using these kinds of empirical cutoffs, we were able to get 97% accurate compared with DNA genotype. So, any questions about that? So I've gone through a lot. I think the next thing that I'm supposed to do is to talk about the portal. But I wanted to stop there and see if you have any questions about the things that I've been talking about. Go ahead.
So what's the advantage of adding internal standard prior to because why do you want to degrade your internal standard which you can control of?
Why in the world would you want to degrade your internal standard? The problem is, in the same way that we can have matrix effects change the amount of peptide that's formed, we can also have differences in the amount of how it's degraded. So it's really for consistency. But if every sample has it before, that peptide will witness the same degradation phenomenon as our endogenous. So we really want the internal standard to mimic the digestion conditions as much as we can, which is why we're having it.
And one more question. After like 4 hours, if you are able to detect the degraded peptide as well as the bound until you are able to detect it, ratio will not change. Like if you assume that in all samples, degradation is same. It's like your control healthy and treatable or like a disease free. So you will still get some difference based on the 4 hour time compared to
So the question I think is, can you tell the difference between diseased and normal, even if you have degradation of the peptide? And the answer is probably yes, because it's all a kinetic process. So if there's more in one sample that experiences decay than the other. And in that respect, you don't need to have internal standards, which is, I think, the essence of your question. So the internal standards, remember, are trying to normalize for a lot of the variability that we have that surrounds digestion. So if there is, it turns out from our experience, if things are degrading at a certain rate, let's say they go too slow because of the matrix effect, then things will actually be generated a little bit slower, and so we can actually begin to overcome some of the variability due to slow digestion by spiking in the internal standards beforehand. But your point is very well taken. If you don't need internal standards, you don't put them in. Internal standards are really to try to control the variability of the assay in some way, but if you don't need to, then it doesn't matter. Any other questions? Yeah.
How do you consider the interference from other proteins or conditions from the patients? Because the lifestyle of the people are different, the gene of the people are different, so it's really a vast variety of the possible situation that interferes with the peptide
Yeah.
target.
Well, there's a program that came from Rudy's lab called SRM Collider, and the concept is SRM space, precursor fragment space, and retention time space is kind of defined. And if you were to throw in the human genome and predict retention time based on hydrophobicity of the peptides, predict the precursor from the peptide sequence, and try to predict some of the transitions that you would see, you can ask the question, for my peptide, what do I expect to see in terms of interferences? We've done those experiments, and they're kind of scary because you type in your peptide and you get a list of 35 things that might interfere. And I really have no way to allay the fears that you're having, other than buying the peptide that you think is most likely to actually interfere, an abundant protein with transitions that actually look like it's near a proline, so that transition might actually exist. Other than getting those peptides and adding them, we don't really have a way to overcome it. So we're still stuck with the blast sequence to make sure that it doesn't look. We do SRM Collider just to see if there's anything that stands out. But usually the proteins are like these weird, like toenail proteins we don't expect to be anywhere else. At least we say that to ourselves. So it's a great question.
We have some kind of black hole in this situation for some of the patients if they have, for example, kidney failure, but the other diseases and this kind of interference of the
Yeah. So the approaches that we always take in the clinical lab to prove that that isn't a problem is to do two things. One is to spike the protein into the matrix, like with patients with kidney failure, which is really disgusting. We have a lot of really bad stuff in there. So spike a protein in and make sure that you get back out where you think you should. That's suggesting that the digestion conditions aren't affected by the chronic kidney disease. The other is to take the sample and dilute it with a human sample, a normal sample, and show that you get the relationship you expect. And then to take that same sample and dilute it with another species. And again, if that's a straight line, both spiking in as well as diluting, you can be more confident that there is no matrix effect and that there's not an interference in one channel or the other. It's not perfect, but it's the best that we can do from the clinical lab perspective. And that's the concept of the parallelism. Any other questions? Okay, well, I had a PowerPoint slide set to talk about the CPTAC portal, but what better way than just to go in and look. So, this is assays.cancer.gov. There's currently 1,500 assays that have been characterized. And if you want to see how they were characterized, sorry, 1,517 now. So we call these characterized assays, and if you see there's a little underline. Yeah, I'm doing it this way. Yeah, it's actually kind of cool. So if you click on the assay characterization guidance document, you can download download. So this is a document that we put together to explain each of these experiments. How to make a response curve by spiking peptides into a digest. How to begin to look at the repeatability, the selectivity, in other words, are there interferences that we might be seeing? What about the stability? Some simple experiments to define the stability of the peptide. And then finally, step 5, which is really the holy grail, which is can I detect the peptide in the sample matrix that I expect to. So literally going through definitions and all of the how the overview and then describing how to go about the experiment in great detail, what kind of mathematical calculations you need to do. Or if you feel so encouraged, you can actually upload your own data and populate the assay portal with your own data if you would like to. So you just have to do a couple of experiments, upload the data, and then actually submit that in the paper or in a grant application that you've contributed to science in that way. So the way that this is set up, so we have proteins and peptides as one column, which laboratory, so UVic, just north of here, up in Victoria, the Fred Putt, a group in Korea, a group, so Steve Carr's lab. Is the peptide modified or unmodified? What kind of assay? Is it a direct MRM assay or is it an immunoaffinity enrichment assay? What is the matrix? So we have plasma and cell lysate. And then we have CBTAC or non-CBTAC. For some reason it was decided that we should make CBTAC different than others. I still don't know exactly why. But if you actually click you can search for your favorite protein. So this is an example. This is HER2. So these assays are all from Hutch and Manny Polowicz's lab. You can see that there are 5 different peptides for this protein. This is an immunoaffinity enrichment step from plasma. The rest are all from cell lines. So if we wanted to click on this peptide, this was an important collaboration between CPTech and the Skyline Group and Panorama. Having the official gene name, other aliases, how long is the amino acid link, how big is the protein, without post-translation modifications, of course. And then here's the whole sequence. You'll notice that this one peptide is actually highlighted in red. And when you put your cursor over the protein sequence, it actually expands. So you can see all of the peptides that have assays in the portal. And there's another way to look at this. This is now the sequence, and there's a color coding. So sequence, isoform, SNPs, and then where are the assays? So these little blue dashes are where the peptides are for each of the assays. So you can click on different peptides. We're already in one of the peptide assays that are over here. So you can potentially click on other things. These are post-translational modifications, specifically phosphorylation events. So you can click out and go visit other websites that describe these phosphorylation events. But that's a polymorphism, sorry. Oh, sorry, those are SNPs. Where's the... Oh, there is a way to link out to phosphorylation. Oh, no. Wait. Can I enable it? Will you be mad at me? It says enable Adobe Flash Player. I'm clicking. I'm allowing.
Go for it.
I did it. Okay.
Perfect.
Nothing bad happened so far. Okay, so here is another view. I'm sorry that it's so bleached out, but here's showing all of the post-translational modifications that are present. This is the domain breakdown for the protein. All of these are being pilfered from other sites. So it's a place where all of this data is getting collated in a single place. But this is really where the power, this is where if somebody comes to you and they say, I want to measure this protein, you could say, oh, let me go check this real quick and see if there's actually an assay that's already been partially validated and at least gives you an idea of making the right step forward. In this case, you'd have to get this is an enrichment MRM, so you'd have to get the antibody, the peptide immunoaffinity step. But most of these antibodies are actually, especially if it's a CPTAC assay, come from the NIH repository, so that the antibody is freely available. All of the assay parameters, so these are required for the upload, what kind of instrument you were on, what was your internal standards, peptide versus protein are actually none, they're actually allowing that now. And then what does the column look like, flow rate? You can actually download the standard operating procedure that's required for upload, so you could go through and figure out. And then there's raw data. What does the chromatography look like? What do the transitions look like? You can actually click to see larger images. This is all being exported from Panorama. Then there's some calculations that are being done. So we're calculating the response curves and getting an idea of what the R squared is around it. Again, all being done in R script and distributed by Panorama, basically looking at different ways to look at each of the data elements. And then there's some summary data about the peptide. What's the lower limit of quantification? You can read how that's defined. What's the limit of detection? Again, you can read how that's defined. And the standard transition nomenclature. So again, all of this data is based on the response curve experiment. And then it's even so nice as to look at the repeatability experiment. So here, instead of doing a 5x5, we propose to do a 3x5 at 3 different concentrations. So now you can see the precision at 3 different levels, and these are each individual transitions. Then the data, again, is summarized down here, so I was able to convince them that it's good to present the total CV of the intra and the inter-assay CV. And now you can see at the lower limit of quantification, you're way above 20% CV. But if you're into that medium, and you can find out what these concentrations are, again, by looking at the standard operating procedure, you can see what kind of concentrations you might be able to expect to see in the matrix of interest. So a lot of interesting data. Down here in the bottom corner, you can actually view things directly in Panorama if you want to, or you can download the standard operating procedure that was uploaded with the assay before it was accepted. So really, this is the goal of the portal was to finally get validation data in the hands of folks like you and me, so that we can look at it and say, do I really want to study this protein or not? So that when that biologist or somebody else walks into our office to see if it's really something that we want to start from scratch, or if it's something that we actually have a starting place, because that's really different. If you already know a peptide that you think you might be able to go after, that's a very different starting place than starting from scratch. So that is the assay portal. If you have any questions or problems with it, let me know.
Is there any instrument dependent situation here for the instrument specification for this data, for example,
is the instrument important? Yeah, I think so.
So is there any information related to the instrument that this data?
Way up top. Where did it go? There we go. So under the assay parameters, it has what the HPLC is, what the column is, what the column dimensions are, and the flow rate. It doesn't talk about the trapping column here, but if you went into the standard operating procedure of it, and it does have this instrument we're working on. So I agree the limit of detection is going to be pretty specific, but hopefully you know how those manufacturers compare with each other. It's not true for every peptide. Yep. So this is what I don't know how to access myself, but I'm going to try. So down here, I believe I'm saying this correctly, in Panorama Web, you can actually start looking at the very raw data that was uploaded with the submission. So you can see every single chromatogram for every single data point that's being presented. So all of the repeatability data, all of the dilution data, you can see the chromatograms in every transition that was used to calculate all of those parameters for each of the peptides. Is that what you mean by raw data? Yeah. Yeah. No, sorry. These are mostly proteins that are thought to be important in cancer, generally defined. I mean, HER2 is really important in trying to characterize breast cancer, but whether or not you can actually use this assay in FFPE tissues, that hasn't been described yet. So no, that isn't it. That's a good question. Yeah, we should be linking out from these proteins to good references that talk about the biology, but that's a little more difficult to do. Okay, if you have no more questions, then I think we're done for the day, Matt, is that right? Yep, we're all done for the day, unless there's special announcements.